All right, let's talk about one of the most fascinating and, frankly, frustrating characters in clinical microbiology, Staphylococcus aureus. For about a third of us, it's just a quiet passenger, living harmlessly in our nose. But for so many others, it becomes a really devastating pathogen. So today, we're going to do a quick briefing. We'll break down its key features, the huge challenge of resistance, and the very different strategies we use to fight it.
Here's the plan. First, we'll get into the dual nature of Staphylococcus aureus. Then, we're going to step into the lab and see how it's actually identified. From there, we'll tackle the big one, methicillin resistance. We'll see how that leads to completely different treatment playbooks around the world. And finally, we'll unpack how aggressive prevention might just be the most powerful tool we have.
So let's start right there, with that duality. Because really, the entire clinical story of Staphylococcus aureus is built on its incredible ability to be both a harmless colonizer one minute and a deadly pathogen the next.
Just think about that for a second. Almost 1 in 3 people are carrying this bug, usually with no symptoms at all. And that massive, healthy reservoir is exactly what makes Staphylococcus aureus such a relentless clinical challenge. It's always there, just waiting for an opportunity. A cut in the skin, a weakened immune system, to flip that switch from a quiet passenger to an invasive threat.
And boy, when it decides to flip that switch, its versatility is just astonishing. It can cause fairly minor stuff like boils, but it's also a top cause of life-threatening bloodstream infections, heart valve infections, and deep bone infections. It can even cause a truly horrific necrotizing pneumonia. And that's not even getting into the toxin-mediated diseases, where its byproducts alone can cause systemic shock. It's quite the resume.
Okay, so with that kind of impact, you can see why fast and accurate lab ID is so critical. Let's walk through the diagnostic process, from the classic methods right up to the latest genomic tools.
Everything, and I mean everything, starts with the gram stain. That classic image of grape-like clusters, that's our first big clue. It's a direct result of the bacteria dividing on multiple planes. In fact, that's where the genus name comes from, the Greek staphylae for a bunch of grapes. This first look under the microscope is so important, it immediately separates it from organisms that grow in chains, like streptococci.
Then, when we grow it on a plate, Staphylococcus aureus gives us even more clues. A lot of strains produce this beautiful golden pigment, which is actually a virulence factor. On blood agar, we're looking for that beta hemolysis, a total clearing of the red blood cells around the colony. And if you put it on mannitol salt agar, it ferments the mannitol and turns the whole medium bright yellow. It really likes to announce its presence.
So here's how it all comes together. The catalase test separates staphylococci from streptococci. Then, the coagulase test has always been the key to tell Staphylococcus aureus apart from its less aggressive coagulase-negative cousins. But today, let's be honest, the gold standards are molecular. A PCR for the NUC gene gives us a definitive species ID, and a PCR for the MEK-A gene is the fastest, most reliable way to know if we're dealing with methicillin resistance.
And this really shows the evolution of our labs. The traditional, phenotype-based methods are still fundamental, but they can be slow. Modern, genotype-based detection is all about speed and precision. PCR can give you an answer in just a few hours. And now, with whole genome sequencing, we can predict the entire resistance profile and track an outbreak with incredible detail, all from one test. It's a total game changer.
And that speed is so important because it brings us to the central challenge with Staphylococcus aureus today, methicillin resistance. This one genetic factor pretty much dictates the entire therapeutic strategy.
So how does this actually work on a molecular level? Well, methicillin-resistant Staphylococcus aureus, or MRSA, carries an extra gene called MICA. This gene codes for a different kind of penicillin binding protein, PBP2A. Think of beta-lactam antibiotics as keys that are supposed to jam the locks, the normal PBPs, to stop cell wall construction. Well, PBP2A is a lock that those keys just don't fit into, so it just keeps on building the cell wall, making that whole class of antibiotics totally useless.
Now, here's a really critical detail for anyone working at the bench. When we test for this resistance on a plate, we use a cefoxitin disc, not an oxacillin disc. Why? Well, it turns out that the MEK-A gene can be a little shy. Its expression can be inconsistent. Cefoxitin is much better at forcing the bacteria to express the gene, so it shows its resistance loud and clear. It just gives us a much more reliable and easy-to-read result.
And these numbers, right from the latest UCAS guidelines, are how we translate those lab tests into clinical action. An isolate is called MRSA if its oxacillin MIC is over 2, or using that cefofoxime disc if the zone of inhibition is less than 22 millimeters. And take a look at the vancomycin breakpoint, also 2. If you get an MIC higher than that, you're in resistance territory, and that has huge implications for treatment.
The threat of MRSA has led to this fascinating split in how doctors practice medicine around the world. Treatment isn't based on one single rulebook. It's all dictated by local epidemiology. It's really a tale of two completely different realities.
And this is exactly why. Look at the Netherlands. Thanks to a really strict national policy, we'll get to that in a minute, their MRSA prevalence is under 2%. Now compare that to many other places in the world where MRSA can make up 20, 30, even 50% of Staphylococcus aureus infections. This one statistic changes absolutely everything about how you approach a patient.
So in a low-prevalence place like the Netherlands, the playbook is incredibly focused. The go-to antibiotic for almost any Staphylococcus aureus infection is a narrow-spectrum beta-lactam, flucloxacillin. Doctors can use it with confidence, knowing the chance of resistance is tiny. This is antimicrobial stewardship at its best, using the most targeted drug you can and saving the big guns like vancomycin only for those rare, confirmed MRSA cases.
Now let's look at the other reality, the high-prevalence setting. Here, you have to assume it's MRSA until proven otherwise. For a simple skin infection, you might start empirically with something like trimethoprim sulfamethoxazole. For a sick, hospitalized patient, you start broad with intravenous vancomycin. But the key here, and this is so important, is de-escalation. As soon as the lab calls and says it's methicillin susceptible, you have to switch back to a beta-lactam. Why? Because drugs like oxacillin or cefazolin actually work better than vancomycin against MSSA.
And of course, treating kids adds another layer of complexity. Everything is dosed by weight. Drugs like flucloxacillin are still standard for susceptible bugs, but clindamycin is often a go-to when community-acquired MRSA is a concern. The big thing to remember is to generally avoid doxycycline in kids under 8 because of the risk of teeth staining. And just like with adults, for severe MRSA, vancomycin is still the workhorse.
So that brings us to the big question. How on earth did the Netherlands get their MRSA rates so low and keep them there? The answer isn't some magic bullet drug. It's a decades-old, incredibly aggressive public health policy that basically redefines what antimicrobial stewardship means.
They call it the search and destroy policy, and it's every bit as aggressive as it sounds. It has 4 main pillars. 1. Search. They actively screen every high-risk patient the moment they come into the hospital. 2. Isolate. Those patients go into isolation immediately, before the results are even back. 3. Destroy. If someone is positive, they go through a standardized decolonization protocol to get rid of it. And finally, trace. If a surprise case ever pops up, they launch a full contact tracing investigation to find out where it came from. And really, this is the core lesson here. The Dutch model proves that the best form of antimicrobial stewardship isn't just about picking the right antibiotic. It's about preventing the need for the powerful broad-spectrum antibiotics in the first place. By literally stopping MRSA at the hospital door, they preserve the effectiveness of their best narrow-spectrum drugs for everyone.
So that leaves us with a really important question to think about. Is this incredibly successful, but also very resource-intensive search and destroy model a blueprint that could be rolled out globally? Or is it a unique success story that's so closely tied to the specific Dutch healthcare system that it would be almost impossible to replicate elsewhere?
We put this briefing together using a Gemini deep search. If you want to go deeper and look at the original studies and data, you can find a complete list of all our sources in the notebook document that goes along with this.
If you found this explainer useful, make sure you subscribe for more briefings on key topics in microbiology and medicine. We know a lot of you work with this organism every day, so we'd love to hear your thoughts. Please leave your feedback and any questions you have in the comments below. Thanks for tuning in.